Hi everyone, this is lecture 13 on vision. And so this is the first on the special receptors. And we're going to find that they're a little more involved and specialized than the general receptors we saw in the last lecture. So we're going to begin with listing the properties of these sensory receptors and the structure and function of the eye, look at a little bit about the histology, and describe the physiology of how the retinal image is formed using terms like refraction and accommodation. You should refer to your textbook because I removed some of the basic slides from this lecture to make it shorter. We'll learn some clinical terms such as emmetropia, myopia, hypermetropia, and presbyopia, as well as astigmatism. We'll look at the receptors called rods and cones and look at the pathway from the eye to the brain at the very end of the lecture. So let's begin by comparing the general versus the special senses, since the special senses have some unique properties. Here's a comparison of the general senses and the special senses. If you notice that the first bullet point is the same for both. The stimuli is processed by the sensory nuclei and transmitted to the thalamus and primary cortex. So it's the same for both. There's nothing different. However, if we look at the second bullet point, we do find differences on the structure of the receptors. In the general senses, if you remember, receptors are endings of sensory neurons. However, in the special senses, most receptors are not neurons, but specialized cells. So we're going to learn some unique cell types for the special senses that detect specific stimuli. But notice that the word most, because there is one that does not follow this. Okay, let's look at this figure here. In the top panel are the general senses, such as touch, pain, and temperature. And we see, as we learned from last time, that the receptive endings of the sensory neurons have the receptors. However, in the special senses, such as taste, light, sound, head movement, we find that there are specialized receptor cells associated with the free nerve endings, which makes them unique. There's one example, however, olfaction or smell, in which the free nerve endings are the receptors. So they're not all the special senses don't have specialized cells, but by far most of them. The exception is being olfaction or the sense of smell. In this next bullet point, we see another difference between the general senses and the special senses. In the general senses, sensation is carried both by axons of both spinal and cranial nerves. However, in the special senses, the special senses are only carried by cranial nerves. Okay, and we can see this in the figure again. In the general senses, axons are carried both spinal or cranial nerves. However, in the special senses, it's only carried by cranial nerves, even in the case of olfaction. And if you recall, cranial nerve number 1 is the olfactory nerve. Okay, so in the special senses, it's only carried by axons of cranial nerves, or in the general senses, it can be either or. Let's begin with the eye anatomy. And the eye anatomy includes both accessory structures and actual specific parts of the eye. And much of this you're going to learn in the laboratory, so I'm not going to go as fine detail as you will hear in the laboratory. The first structure is the palpebrae, and this is a good term to know because these are your eyelids. Okay, and they're composed of 2 thin folds. And as you might expect, they're there to protect the eye and help distribute tears when we blink. Okay, so here I've indicated the upper eyelid and inferior eyelid, superior and inferior eyelids. Okay, I think most of those is fairly straightforward. Your eyebrows and eyelashes include hairs that protect the eyes by blocking perspiration, as in eyebrows. That's why we have eyebrows. And light. That's why we have eyelashes. And eyelashes are also innervated to trigger blinking. Okay, so you can have a reflex by poking someone in the eye and they'll blink. Okay, and so here are eyebrows and eyelashes. So these two structures, I think, doesn't have to be explained any further. Okay, here's something you might not have heard about: the conjunctiva. This is the epithelial membrane lining the posterior surface of the eyelid, so it goes underneath the eyelid and also around the surface of the white part of the eye. Okay, if you have children, many children suffer conjunctivitis or pink eye when it gets infected. So we could see here that there's a palpebral conjunctiva as well as a ocular conjunctiva. Okay. And so here it's underneath the eyelid, and this is on the surface of the eyelid, surface of the white part of the eye. Another structure you may have heard of, but may not discussed at length, is the lacrimal apparatus. The lacrimal apparatus produces the tears and helps drain the eye. The lacrimal gland secretes the tears and mucus to lubricate and wash away dust and debris. Now, if you recall from SCB 203, you learned the lacrimal bone, and there was a little hole in the lacrimal bone that you use for a landmark. And that hole is the area where the lacrimal apparatus will drain the tears into your nasal cavity. So if you experience when you cry a lot, your nose starts running, it's because the tears are draining into your nasal cavity. And here is your lacrimal gland, I guess the superior lateral portion of your eye. Your muscles are innervated by extrinsic eye muscles. They're extrinsic, meaning they're outside the eye, and their function to move the eyeball within the socket. And there are 6 of them. 4 are rectus muscles, meaning straight, and 2 of them are oblique muscles, and then they have curved fibers. So there's superior, inferior, lateral, and medial rectus muscles, and the superior and inferior oblique muscles. You should know what they do in your eye. And so here you can see the different muscles in this figure. So we have the lateral rectus muscle here. We have the superior rectus muscle, the medial rectus muscle, and the inferior rectus muscle. And they're inserted based on their name. We also have the superior oblique muscle and the inferior oblique muscle. Okay, and here's a lateral view. So you can see the lateral rectus and medial rectus muscle. In the next table describes the control of these muscles and what they do. Okay, and so what's nice in this figure is in words they provide you their function, and on the right here they show a person's eye indicating what they actually do. So the superior rectus muscle elevates and turns the eye medially. The inferior rectus muscle depresses and turns the eye medially. The medial rectus muscle, well, this moves the eye medially. The lateral rectus muscle moves the eye laterally. The superior oblique muscle moves the eye downward and laterally. And the inferior oblique moves the eye upward and laterally. Okay. We won't mention the lateral palpebra superalis muscle. Okay, this is your eyelid. Now let's begin talking about the anatomy of the eyeball itself. Okay, we need to do this so that when we describe the physiology of the eye, you'll be familiar with the terminology. The eyeballs consist of different layers. It consists of the lens. It has different cavities and chambers. So let's begin looking at the anatomy of the eyeball. There are 3 layers or tunics of the eyeball. There's a fibrous layer, a vascular layer, and a neural layer. And this figure here shows you the 3 layers. The fibrous layer, okay, the vascular layer, and the neural layer. So there are 3 layers to the eye. Let's look at each one now. Let's look at the fibrous layer. And this is the most superficial, the outermost layer. And it's composed of 2 structures of the eye, the sclera and the cornea. Okay? So you can see from this picture here, the anterior part of the eye is the cornea, and the majority of the eye is surrounded by the sclera. And so the sclera is the white part of the eye and composed, as we said, majority of the eye. 5/6 of the eye is covered by the sclera. And it's white and opaque because due to many irregular collagen fibers that maintains the shape of the eye. So it's there so the eye maintains its shape. And you can see the sclera here. The cornea is continuous with the sclera at the anterior of the eye. However, it is translucent, not opaque, and is due to parallel collagen fibers, very little water, and it is avascular, meaning it has no blood vessels. It's translucent because it's important for admitting and focusing light into the eye. So it's going to play an important role in the physiology of the vision. So here is the cornea in the anterior part of the eye. So the fibrous layer is composed of the cornea and the sclera. The second layer is the vascular layer, and it's beneath the cornea and the sclera. So it's going to be the middle layer. And it's composed of 3 structures, the choroid, the ciliary body, and the iris. So in this picture we see the choroid, ciliary body, and the iris. Okay, here's the choroid. The choroid, the ciliary body, and then the iris. Let's go through each one briefly. The choroid is highly vascularized, giving the layer its name. And it's also pigmented to reduce scattering of light, so it helps keep the eye dark. The ciliary body is continuous anteriorly with the choroid, and it's composed of 2 structures, smooth muscle and the suspensory ligaments. The suspensory ligaments helps support the eye. Okay, so here is the ciliary body of smooth muscle, and then we have the suspensory ligaments that's positioning the lens. The iris is the pigmented part of the eye, and it's anterior to the ciliary body, and in the center has an opening called the pupil. Okay. The iris is pigmented due to melanin that helps determine eye color, and it contains smooth muscles that controls the amount of light entering the eye. And in lab, you might be talking about the pupillary reflex. And so here's your pupil, the opening of the eye. If it's exposed to increased light intensity, under the parasympathetic nervous system will cause it to constrict to protect the eye. If you have less light and you're in a dark environment, your sympathetic nervous system will reflexively cause it to dilate to open the eye. Okay, so parasympathetic is for constriction of the pupil. Sympathetic is for dilating the pupil. The third layer, and innermost, is the neural layer, and it's composed of the retina. And the retina contains the receptors and also the neurons that will send the images to the brain. So it contains the photoreceptor cells that detects light and produces the neural signal. So here is the neural layer containing the retina. Okay. And if you saw pictures of a retina, it looks almost, it's easy to lose because it's sort of brownish color or beige color. Now the retina, as we said, contains the photoreceptors. There's an area in the retina that contains the highest amount of the photoreceptors, and this is called the fovea centralis. Okay, it's in the center of a yellowish region called the macula lutea. So figure here, we see the fovea centralis within the area of the macula lutea. Okay, I realize this is yellow too, but that's another structure we'll talk about momentarily. Also on the retina is an area called the optic disc, because this is where the axons of the optic nerve join to form the optic nerve. And this particular area does not contain any photoreceptor. Because of this, it is known as the blind spot. Okay, so this yellow layer here is the optic disc, where there are no photoreceptors. Now, it is called the blind spot. However, it is not normally noticeable because your brain fills in the missing information based on the images next to it. So your brain kind of fills in the blind spot so we don't notice it. However, there are different tests you can do to demonstrate the blind spot. Of course, you can't do it from these PowerPoint slides normally, but you can test these on a piece of paper, for example. So to test your left eye, you cover your right eye and focus on the X. Okay, and slowly move your book toward or away from you until the dot disappears. When the dot disappears, that means the image is focused on your blind spot. Then you can do the other eye by testing your right eye and covering your left eye and focusing on the dot, and then the X will disappear. Another way of testing your blind spot is using this figure. To test your left eye, cover your right eye and focus on the X. Slowly move your book toward or away from you until the pencil disappears intact. So when the image hits your blind spot, your brain will fill in the pencil because it assumes what it's going to be there is going to be the same on either side. To test your right eye, turn the figure upside down and cover your left eye and repeat the process. The next structure of the eye is the lens, and it's a flattened sphere posterior to the iris and pupil that focuses light on retina. It's bulged on either side. Okay. It's surrounded by the ciliary body and is connected by the suspensory ligaments. Now, you might have heard of a cataract. A cataract occurs when the lens becomes cloudy, and oftentimes they can just remove the lens, and you can see normally. Okay, so here's the lens, and you can see the suspensory ligaments supported by the ciliary body. Now, in these figures, it looks like it's superior and inferior to the lens, but keep in mind that this is only a section of the eye, and it's actually going completely around the lens. Okay, so if you looked at the anterior of the eye, it looks like a circle. The eye is divided into different cavities and chambers. Okay, the lens and ciliary body divides the eye into the anterior and posterior cavities. So anterior to the lens and ciliary body is the anterior cavity. Posterior to the lens and ciliary body is the larger posterior cavity. Okay, the anterior cavity is smaller and is composed of a fluid called aqueous humor, and it's further subdivided by the iris into the anterior cavity and then the anterior chamber and the posterior chamber. The larger posterior cavity is filled with a more jelly-like structure called the vitreous humor that helps maintain the shape of the eye. When you dissect the eye, it comes out like a little blob. So we can see this in this figure here. So the lens and the ciliary body divide the eye into the anterior cavity and a posterior cavity. In the anterior cavity is composed of the aqueous humor. The posterior cavity is composed of the vitreous humor. Now the iris divides the anterior cavity into an anterior chamber and a posterior chamber. So let's begin to show how light is focused on the retina. Because remember, the retina has the receptors for vision. Okay. So when light strikes a non-transparent object, it causes the light rays to diverge away from the object. Okay. So this is sort of basic, the principle of using a microscope, because the object absorbs light and you're looking at the reflected light from that object. But in the form of the retina, if the object is more than 20 feet away, the reflected rays are nearly parallel because they're so far away, it's coming toward the eye almost at parallel lines. However, if the object is very close, less than 20 feet, the rays are still spreading out or diverging. Okay, so as you might expect, in order to focus this image, the eye is going to have to bend or refract these light rays onto the fovea centralis. It doesn't have to work so hard in objects that are far away because they're almost parallel. Let's take a look at a picture of this. Okay, so here's an image of an apple. Okay, at more than 20 feet, the light rays are almost parallel, and so it doesn't take much effort to bend these onto the fovea centralis. However, looking at objects less than 20 feet, these are still diverging, and the eye will have to make extra effort to bend these light rays onto the fovea centralis. Now, when you're comparing the eye and the cornea, the lens and the cornea, we always put much emphasis on the function of the lens. But as we said, if you damage your lens, it can be removed and you can still see normally. Because the greatest degree of refraction that occurs from light into air is through the cornea. And so the lens does add some refraction, but does not as much as the cornea. So if you have to have your lens removed, you should still be able to see fine if your cornea is normal. So let's look at light rays from distant objects. Remember, they need little refraction to focus on the retina since they are nearly parallel when reaching the eye. And the cornea does most of the refraction, bending the light rays toward the fovea centralis. Okay. Of course, the flattened lens provides the rest, because the lens can stays flattened so that it doesn't need to bend. Is not required to bend the light rays or refract them significantly. Okay, so we're looking at this tree, and we're looking at it from a distance. So the light rays entering the eye are almost parallel. The cornea does most of the refraction, and the lens helps a little bit. And so now, in perfect vision, the image is focused on the fovea centralis. Okay, remember here is the blind spot where the nerves are forming the optic nerve. The neurons are forming the optic nerve. Now, if this is done properly, we have what's called the emmetropic eye, or emmetropic state of the eye, and this is the normal eye. So in this figure here is the normal eyeball shape. And if it's perfectly normal, then the light rays will be refracted right on the fovea centralis. Okay, and that's the emmetropic eye. Okay. Now, objects that are very close to the eye, the light rays are still diverging, and the cornea needs help. They cannot bend the light rays themselves onto the fovea centralis. So it requires the assistance of the lens. Okay, and it increases the refraction by becoming rounder. Okay, so a rounded lens will bend the light rays more than a flatter lens. And this process of the lens becoming thinner and thicker is called accommodation. Okay, so by the accommodation of the lens, it can then refract light to a greater extent. So here we're looking at the apple at a very short distance. The light rays are diverging. The cornea tries to bend them, but cannot bend them enough to focus on the fovea centralis. So the lens rounds up so that now they're bent to focus on the fovea centralis. Okay, so remember, at distant objects, the lens can stay flat, but in close objects, the lens can round up for near vision. So the question is, how does the lens change shape? Well, distant objects, the ciliary body relaxes, moving ciliary body away from the lens. This will increase the tension on the suspensory ligaments and flattens the lens to decrease refraction. Now, when I was a student, I always had a hard time understanding this, because if you, I just could not, I read the words, but I could not visualize it. Okay, so in this figure here is showing that the choroid ciliary body is actually a circular structure with suspensory ligaments and attached to the lens. So in this lateral view, we see that the eye is curved. Okay. So when the ciliary body relaxes, it moves away from the lens. So it's going this way. Okay. But notice it's wider here. And so it's wider here, it's going to apply more tension on the suspensory ligaments, and the lens will get flatter. And once this figure helps me explain the previous slide in words much clearer. That's how the ciliary body can relax but flatten the lens, because of increased tension on the suspensory ligaments, because there's a further distance back here than it is up here. So during accommodation, the ciliary body contracts and moves closer to the lens. This decrease in tension on the suspensory ligaments allows the lens to round up and increase refraction to focus on nearby objects. The lens by nature wants to round up. Okay, if you took the lens out of the eye, it'll be like a little ball. So if the ciliary body contracts, it's going to move closer to the front of the eye. If it does this, the distance in the front of the eye is shorter than in the back of further back in the eye. As a result, the tension on the suspensory ligaments are going to be relaxed, and the lens will round up for close-up vision. So this explains why contraction of the ciliary body removes tension on the suspensory ligaments, and the lens rounds up. Of course, sometimes this doesn't always happen, and it may also be due to an irregular shape of the eye. And this is when we get these disorders called presbyopia, hyperopia, and myopia. We have what's called the near point of accommodation. So if an object is too close to the lens, it cannot round up enough to provide refraction. And so we have what's called the closest point at which we can focus an object is the near point accommodation. And of course, this increases with age. So if you have elderly relatives or friends, you might see without their glasses, they hold a book or a newspaper, far away from their eyes to be able to read it, because they can no longer accommodate close-up vision. And this increases with age. And this term is presbyopia. Presbyopia is the age-related condition when the lens stiffens and the near point is about 10 to 20 inches and close vision becomes difficult. Okay. How about hyperopia, which is farsightedness? It can be due to too short of an eyeball, or can be too flat of a cornea. And the lens cannot accommodate an image focuses behind the retina, and the image focuses behind the retina. So somehow you need to refract the light more so that the image lands right on the fovea centralis. As a result, close-up objects are blurry, but you can correct it with a convex lens. So this figure shows hyperopia or farsightedness, where the cornea of the lens cannot refract the light onto the fovea centralis, and actually it converges behind the eye, and it could also be too short of an eye. But you can correct this with a convex lens. And the convex lens will assist the cornea on the lens to refract the light right onto the fovea centralis. Likewise, we have myopia, which is nearsightedness. And this can be too long of an eyeball, or the cornea curves too much. And as a result, the lens cannot flatten enough and the image focuses in front of the retina. Okay. As a result, distant objects become blurry and can be corrected by a concave lens. So here in this figure, we see this eye. It could be due to too long of an eye or imperfect curvature of the cornea. And as a result, too much refraction of the light causes the image to focus in front of the fovea centralis. However, by using concave lenses, this diverges the light rays so that they converge right on the fovea centralis. And this is myopia or nearsightedness when you have trouble seeing at far distances. Okay. You might have heard of an astigmatism. An astigmatism is an irregular curvature of the lens or cornean rays, and they're not evenly refracted. Okay. Let's go to the retina now, because this is where the photoreceptors are, as well as the neurons for detecting light. Remember, this is the inner layer of the eye. There are two types of photoreceptors in the retina, and they're named by their shape. They are the cones and the rods. So in this section of the retina, on the left side is an illustration, and on the right side is actually a photomicrograph. We see the photoreceptors. What's interesting is that the photoreceptors are the furthest away from the image. They're very near the back of the eye. And of course, we see the choroid layer in the next layer over. Okay, so we have the rods and we have the cones. Okay, the cones contain pigments for color vision. So this is kind of easy because color starts with a C and cones start with a C. And they work best in bright light. And you have about 5 million cones, and most of them are highly concentrated in the fovea centralis. So that's why we want the refraction to be right on the fovea centralis. The rods cannot detect color, but are sensitive in less intense light. Okay, so they're better in darker light, or less light. And you have about 100 million rods, but none of them are in the fovea. And they increase further away from the fovea centralis, and then they taper near the edge of the retina. Okay, so here's a micrograph, a scanning micrograph of the rods and cones. Okay, and we see that they absorb at different wavelengths. Okay, the rods and cones synapse with other cells called bipolar cells that then communicate with the retinal ganglion cells. Then the axons of the retinal ganglion cells are those that form the optic nerve, or cranial nerve number 2. Okay, so we have here are the rods and cones. They synapse with bipolar cells that communicate with retinal ganglion cells, and these form the optic nerve. Okay, there are also other cells called horizontal and amacrine cells that are also involved in the imaging process to make it a more sharper image. And we'll mention those a little bit later. Horizontal cells form connections between photoreceptors. Amacrine cells are along dendrites of retinal ganglion cells. So here are the horizontal cells. As you might expect, they're horizontally situated in the retina. And then we have the amacrine cells. Okay, let's look at the structure of the photoreceptors. And these are the rods and cones. And of course, they're right next to the pigmented layer, which is the choroid layer. The outer segments contain discs that absorb light. Okay. And the inner segments contain mitochondria and organelles to make new discs. So they're just uniquely shaped cells. Okay, so both the rods and cones is an outer segment and an inner segment where there's mitochondria. They have a cell body and then synaptic terminals. So just like they're kind of like torpedo-shaped cells. The rods, the outer segment is cylindrically shaped and contain about 1,000 discs. Some people have made an analogy. They look like a container of coins. A sheet of a stack of coins. Each disc contains pigment rhodopsin in its membrane. Rhodopsin itself is composed of a protein called opsin and a pigment called retinal, which is derived from vitamin A. Okay. So here's rhodopsin. It's composed of opsin and retinal. And these are in the discs of the rods. The cones are, like you might say, have cone-shaped, and their discs contain the pigment iodopsin. And iodopsin is composed of the protein photopsin, and again, retinal. Okay, so here are the cones. Okay, they just don't have a figure like this for the cones. Okay, the cones forms the three forms of photopsin absorb at wavelengths in blue, green, and red ranges, and the perception color is generated by the varying combinations of cone populations. And so by the different cone populations, you can see the different spectra of the different colors. Now let's look at the transduction of the photoreceptors. Okay. Now this is where this is our first receptor where they behave not like we normally think of receptors should. Okay. In the photoreceptor cells, they are depolarized already in the dark and continue to release neurotransmitters. Okay. Upon light, it hyperpolarizes the photoreceptors. So this is kind of our first time where we have things kind of backwards than what we normally have seen, and reduces the neurotransmitter release. So let's look at what happens in the dark first. Opsin and cis-retinal combine to form rhodopsin in the disc membrane. Okay, and this similar actions are occurring in the in both the rods and the cones. Okay. A G protein complex called transducin and an enzyme called phosphodiesterase are inactive, and we'll talk about these in a little bit. For now, we're just introducing the terms. So remember, transducin is a G protein, and an enzyme is called phosphodiesterase, and they're both inactive in the dark. So here is our picture of transducin. Here's rhodopsin with cis-retinal. Okay. Here's phosphodiesterase. Both transducin and phosphodiesterase are inactive in the dark. But remember, in the dark, the ion channels are continually allowing sodium to enter the cell, keeping it depolarized. Okay. And sodium channels opened by the second messenger called cyclic nucleotide monophosphate, also known as cyclic GMP. In this figure here, we see that the sodium channels are in the dark are always open, allowing sodium to enter, depolarizing the membrane, and they're opened by the second messenger called cyclic GMP. Okay. Now, what happens in the light? When light strikes rhodopsin, it causes retinal to separate from opsin. Okay. And retinal changes from a bent form called cis-retinal into a more straighter trans-retinal shape. So cis is bent, trans trans is the straightened retinal, and it changes upon striking of light. Okay, so cis retinal, upon light striking it, turns into transretinal and separates from opsin. So now opsin changes its shape once the cis retinal leaves, triggering a set of reactions to activate transducin, which then activates phosphodiesterase. So the retinal leaves after changing shape, opsin changes shape, interacts with transducin, activating it, and this separates to activate the enzyme phosphodiesterase. Now, the activated phosphodiesterase converts cyclic GMP, remember this was keeping the sodium channels open, into GMP. As a result, cyclic GMP decreases to keep the sodium channels open. So without cyclic GMP, the channel closes and reduces the influx of sodium. And as a result, the membrane hyperpolarizes. Okay, so here we have transducin interacting with the enzyme phosphodiesterase. Phosphodiesterase will convert cyclic GMP, which was keeping the channel open, into GMP. With the loss of cyclic GMP, the sodium channel no longer stays open and closes, and the membrane hyperpolarizes. Because what they're not showing here are other channels like potassium channels that are still open. When transretinal dissociates from opsin, rhodopsin is said to be bleached. And once these photopigments are bleached, they cannot respond to light until it reassembles again. So the retinal and the opsin has to all be put back together again. So this comes to the explanation of dark and light adaptation. Okay, and dark and light adaptation is also involved in the pupil size. Remember when we said pupils constrict or dilate? Okay, and we said that in bright light the pupils will constrict and they'll dilate in low light. However, if you notice that it takes longer to adjust to dark light than to bright light. So for example, if you're going to a dark movie theater, it takes longer for you to adjust to the light than after the movie in the daytime when you go back to the sunlight. So in dark adaptation, all the rhodopsin are bleached and unable to function because they are coming in from bright light. Okay. When light is reduced, the cones can't function, and rods are slow to regenerate enough rhodopsin to function. And it may take about 40 minutes for dark adaptation to occur. The reason why it takes longer for dark adaptation is because the rods are very slow to regenerate enough rhodopsin from bleaching. In light adaptation, we know that the dark rods are fully functional, but bright light bleaches the rods and cones, resulting in a blinding glare. The rods become non-functional as rhodopsin is bleached as fast as it is generated. However, the cones, they regenerate pigments much faster than the rods, and only in a few minutes. So going back into the bright sunlight, adaptation is much faster because the cones can regenerate their pigments quicker than the rods. Okay, let's look at the image processing by the retina. Okay, remember the rods and cones are associated with bipolar cells and then the retinal ganglion cells, and these form the optic nerve. Okay, so let's think about this. There are more than 105 million photoreceptors, but only 1 million retinal ganglion cells, and the photoreceptor cells have to synapse with the ganglion cells. So this, by looking at this, you know that there's going to be conversion occurring. A whole bunch of photoreceptor cells converging onto a few million retinal ganglion cells. However, the image is sharpest when the ganglion cells receives input from only one photoreceptor, as in the fovea centralis. So you have this divergence outside the fovea centralis. But in the fovea centralis, the image is sharper because you have more of a one-to-one relationship between the photoreceptor cells and the ganglion cells. So, in the dark, here is a photoreceptor. It's associated with a bipolar cell, which is associated with a retinal ganglion cell. Okay, so in the fovea centralis, it'll be more of a one-to-one relationship. Okay, and we see that the photoreceptor depolarizes and releases glutamate onto bipolar cells in the dark. Okay, remember that it's not inhibited by the phosphodiesterase. The neurotransmitter binds to receptors, and this neurotransmitter is glutamate. And the glutamate binds to the receptors and inhibits the bipolar cell, reducing its release of neurotransmitters. Okay. In this case, the neurotransmitter is inhibitory, and so there is no action potential to the retinal ganglion cells. When we look at the dark in the light, remember the photoreceptors now, the pigments are being bleached. This cis-retinal is leading to the trans-retinal. Opsin changes shape. Okay. And as a result, the sodium channels become inhibited, and so the cell actually hyperpolarizes, and it stops releasing glutamate. As a result, the inhibitory action of glutamate on the bipolar cells is no longer there, and so now the bipolar cell can now depolarize and release its neurotransmitters to the retinal ganglion cells to produce an action potential. Okay. We mentioned the horizontal and amacrine cells, and we said and their function is to alter the sensitivity and increase the contrast of objects. However, their role is not as clear as in the rods and cones. Finally, let's look at the visual pathway. So the information must be sent to the brain for conscious awareness and interpretation. Okay, and so we have what's called the visual pathway. The visual field refers to what is seen with both eyes open. So we have a right eye and left eye. And with each eye, it's divided into right and left visual fields with a focal point in the center. And the visual fields overlap between the two eyes, giving us binocular vision. So here's the right visual field. Here's the left visual field. And the focal point's in the very center with both eyes. Okay, so we have our right eye and we have our left eye. Okay, and so what happens is the retina of each eye detects visual stimuli from portions of the right and left visual fields. Okay, if you notice that the optic nerve forms what's called the optic chiasma, where it forms a little X-like, and some of the fibers from the right eye continue on to the right side, but some of them cross over at the optic chiasma to the left side. Likewise, some of the optic fibers from the left eye stay on the left side, but some of them cross over to the right side at the optic chiasma. So that the processing of the image is handled, some of the parts of the image is handled by the opposite hemisphere of the brain. And this helps put together a binocular image. Finally, remember the occipital lobe was where the primary visual cortex is, and after passing through the thalamus, it goes to the primary visual cortex in the medial portion of the occipital lobe. Okay, and I do go through each one here. So here we see the retina of each eye detects the visual stimuli from their visual field. Here, some of the fibers cross over to the opposite cerebral hemisphere and enter the thalamus. Remember, this crossing over is the optic chiasma. And finally, the visual stimuli travel from the thalamus to the primary visual cortex in the medial portion of the occipital lobe. And so the visual pathway, this summarizes what we just talked about. The cornea and the flattened lens provide enough refraction to focus light rays on the retina, okay, because the object is more than 20 feet away. Your photoreceptors hyperpolarize, and retinal ganglion cells send action signals to the optic nerve. Selected axons cross from the optic chiasma so that stimuli from each half of the visual field are sent to the opposite hemisphere. The primary visual cortex provides conscious awareness and initial processing of the shape, color, movement, and each hemisphere processes stimuli from the opposite eye's visual field and puts the two visual fields together.